Welcome to Parasitology. Today, we are diving into the fascinating and dangerous world of parasitic protists. Imagine your body as a beautifully furnished mansion, and suddenly, invisible squatters move in, eating your food and damaging the walls. These microscopic hijackers are responsible for some of the most devastating diseases in human history. By the end of this lecture, you will understand exactly what they are, why they are so hard to defeat, and how medical professionals use mathematics to track and fight them. Let us begin our journey into the microscopic world. Building on that mansion analogy, let us define exactly what these invaders are in biological terms. Parasitic protists are single-celled eukaryotic organisms. This means their entire existence is confined to just one microscopic cell. Yet this single cell is a complex machine, capable of eating, reproducing, and evading your immune system. The term parasitic means they have adopted a lifestyle of absolute dependency. They cannot survive on their own. They must live in or on a host organism. They derive all their nourishment at the direct and harmful expense of that host, leaving the host severely sick. Now that we know they are single-celled, we must focus on the word eukaryotic, because this is what makes them so incredibly dangerous. Unlike bacteria, which are simple and primitive, eukaryotic cells have a true membrane-bound nucleus that holds their DNA. Here is the terrifying part. Human cells are also eukaryotic. Because these parasites share a similar cellular machinery with us, treating them is incredibly difficult. Drugs designed to destroy the parasite's complex internal structures often carry a high risk of damaging the host's own cells. It's like trying to poison a thief who shares your exact biology. Understanding their biology leads us to why we dedicate an entire field of study to them. These microscopic creatures are not just a biological curiosity. They are responsible for massive global suffering. For instance, the protist Plasmodium vivax causes malaria, a disease that affects hundreds of millions of people annually and claims hundreds of thousands of lives, especially young children. Another notorious example is Entamoeba histolytica, which causes severe amoebic dysentery. By studying how these foreign cells evade our highly advanced immune system, scientists can develop targeted drugs and vaccines to prevent massive outbreaks and save countless lives. Before we look at how to measure these diseases, let us visualize exactly what this microscopic hijack looks like. On the left, you see a single parasitic protist. Notice its distinct cell membrane and the dark nucleus in the center, highlighting its complex eukaryotic nature. The arrow shows this parasite invading the host. On the right, we see the result: a human red blood cell that has been completely hijacked. Inside, you can see multiple new protists multiplying rapidly. This single image perfectly captures why the host becomes so severely ill, as their cells are destroyed from the inside out. Now that we have seen how a single red blood cell is invaded, we must learn how to measure this battle across the entire bloodstream. This brings us to a critical mathematical tool called parasitemia. This formula allows doctors to quantify the severity of the infection. Conceptually, it simply calculates the fraction of your blood cells that have been hijacked by the parasite and converts it into a percentage. P stands for parasitemia. I is the number of infected red blood cells counted under a microscope, and T is the total number of red blood cells in that same sample. If the infected count increases while the total remains constant, the parasitemia percentage rises, indicating a worsening infection. Let us see this formula in action with a real clinical scenario. A patient arrives at a rural clinic with a dangerously high fever. A technician takes a blood smear and counts a total of 500 red blood cells. Out of these, 25 cells contain the Plasmodium parasite. We need to find the parasitemia percentage. We substitute our known values into the equation, dividing the 25 sick cells by the total sample size of 500. This fraction simplifies to the decimal 0.05. Multiplying by 100 gives us a final parasitemia of 5%. In a medical setting, a 5% parasitemia indicates a severe, life-threatening malaria infection requiring immediate intervention. We've just seen how to measure the severity of an infection in a single patient using parasitemia. But what happens when an entire village gets sick? Doctors treat individuals, but public health officials must treat populations. To track dangerous outbreaks and identify the source of a disease, epidemiologists need a different metric. They need to know what proportion of an entire community is currently harboring the parasite. This shift in focus from the microscopic blood cells of one person to the macroscopic view of a whole population requires us to adapt our mathematical approach. This brings us to a crucial epidemiological metric known as the prevalence rate. Conceptually, it is identical in structure to the parasitemia formula, but the scale is entirely different. Instead of counting cells, we are counting people. P-rev stands for the prevalence rate as a percentage. The variable I now represents the number of infected individuals in a specific area, and T represents the total human population of that area. If the number of infected individuals goes up in a fixed population, the prevalence rate increases, signaling a spreading outbreak that requires immediate public health intervention. Let us apply this prevalence formula to a public health emergency. A medical team is investigating an outbreak of amoebic dysentery in a village with a total population of exactly 8,000 people. After testing, they find that 320 individuals are actively infected with Entamoeba histolytica. To find the prevalence, we place the 320 sick people over the total population of 8,000. Dividing these numbers gives us the raw decimal proportion of 0.04. Multiplying by 100 yields a final prevalence rate of 4%. A rate this high strongly suggests a contaminated communal water source is spreading the disease. Now that we have calculated a 4% prevalence rate, we must understand what this number actually means for the community. Data alone does not save lives. The actions driven by that data do. When public health officials see a prevalence rate this high for a waterborne parasite, it acts as a massive red flag. It provides the hard, undeniable evidence needed to justify emergency funding and resources. Authorities can immediately bring in water purification systems, isolate the contaminated sources, and begin treating the sick, effectively halting the microscopic hijackers before they infect the rest of the village. We have covered a tremendous amount of ground today, so let us summarize our key takeaways. First, we learned that parasitic protists are complex, single-celled eukaryotic organisms that survive by exploiting a host. Second, because their cellular machinery is so similar to our own, they are brilliant at evading our immune defenses and are incredibly challenging to treat safely. Finally, We saw how mathematical tools like parasitemia and prevalence rates allow medical professionals to accurately measure infection severity and track dangerous outbreaks. Mastering the biology of these microscopic hijackers is our absolute best weapon in the ongoing fight to protect global public health.